Okay, today I'd like to continue our discussion of DNA technology and talking about some different examples of technology and how they're utilized. So the first one I want to discuss is something called polymerase chain reaction, or PCR. And the purpose of PCR is to make copies of a gene. And so it's a pretty simple process in that it involves a cycle of heating followed by cooling. And then these alternate over and over again to produce our copies of genes. So it's in that heating and cooling cycle, it's a three-step process. And so I'll start with this, then we'll look at some diagrams. But the steps are denaturation. And so denaturation will occur at the peak temperature, about 94 to 96 degrees. And this causes the separation of the two strands of DNA of interest to us. And then in the cooling phase, there's initially what's known as annealing, where a short segment of RNA will recombine with the strands of DNA. And then extension would be where DNA polymerase then extends each strand of DNA to get us back to a two-stranded piece of DNA. So let's go ahead and look at some diagrams that kind of illustrate this for us. But before we get to that, what we would do then in this PCR process is that the PCR cycler basically then is like an oven. It will heat and cool. And so in one of the tiny vials that we utilized in our last module, what you'd do is put some sample of DNA, and that DNA sample then would have the gene of interest to us. And one of the good things about PCR is that we don't need to know exactly where those genes are, but what we do need to know is some of the sequences on either side of our gene, and we'll see that in a couple of seconds. So anyway, your DNA sample of interest would have to go in this little vial, and then the nucleotides that we would need to build new DNA strands. And of course, our 4 nucleotides would contain adenine, guanine, cytosine, and thymine if we were making DNA. And then we'll also need some short RNA primers, and we'll see why we need those in a second. And then also a type of polymerase that we call Taq polymerase. And Taq polymerase is a heat-tolerant DNA polymerase. It comes from a heat-tolerant bacteria known as Thermus aquaticus, and that's where the Taq part of this comes from. So again, it's a DNA polymerase that tends to work at higher temperatures than DNA polymerase normally does. Now, in the process itself then, what we would start is with a chromosome. And of course, this is not to scale. The chromosomes are really long. But we would be interested in a particular gene in this chromosome. And so we'd be targeting a particular gene. But again, what's important about PCR isn't so much where that gene is, but what we would need to know is some of the sequences on either side of the gene. Okay, and I think that'll become obvious here in a second. So anyway, again, we go through the heating cycle in which the DNA double helix will separate and the strands separate. So the denaturing of the chromosomes will occur, and again, this is separation of the DNA strands. Then in the cooling phase, initially what would happen is that RNA primers, short pieces of RNA, would reanneal with complementary bases. And so again, this is why, even though we may not know everything we want to know about the gene, we do need to know some of the sequences on either side of our gene, because we have to produce primers that are complementary to the bases on either side of our gene. And then they will anneal with those strands of DNA. Now, this is an important step, because you may recall that DNA polymerase cannot bind to a single strand of DNA. So again, what we want to do here is produce complementary strands of DNA so that we end up with 2 copies of our chromosome or 2 copies of our gene, as opposed to having 1 copy. So DNA polymerase requires a double strand to bind to, so that's the purpose of the RNA primers. We need the primers there. They will anneal with the complementary strands. That then allows DNA polymerase to bind to those double strands. The DNA polymerase can then build new strands 5 to 3, and at the end of the process then we will have 2 copies of our gene at the end of a 1 heating and 1 cooling cycle. And then again, it's a very simple process then, so still leaving that stuff in the vial then, we just need to go ahead and continue the heating and cooling cycles to make more copies of our genes. And so now we've got 2 copies of our genes. We'll heat and cool again. So the heating process will cause denaturization. In the cooling cycle, we'll get reannealing. The DNA polymerase will bind, and then extension of the strands. And that will double our copies of genes again. So what happens then in this process is that with each heating and cooling cycle, we effectively will be doubling the number of copies of genes that we have. And again, there's some important uses to this, but you might be wondering, why do we need a lot of copies of genes? And I'll get to that a little bit later. So we do have some situations where we would want to use multiple copies of genes. Now, one of the applications of PCR is that we can actually use this in concert with gel electrophoresis to produce DNA fingerprints. And so this is especially useful when you don't have a big sample of DNA, when you have just a small amount of DNA, and you might not be able to run a traditional DNA fingerprint, as we discussed in the previous module. So what you can do in those situations then is that you can, again, target a particular gene. We can run PCR, but what we would be targeting here is not just any gene. What we'd be interested in targeting would be VNTRs. So what we could do then is target specific VNTRs, and then produce primers on either side of those VNTRs, and we could make multiple copies of those VNTRs, specific VNTRs. And then, again, if we target several different VNTRs, what we would do is be producing PCR fragments instead of restriction fragments, if you will. So then we could go ahead and load our samples into a gel. We could go ahead and run our gel. And again, the nice thing about this is that when we run a traditional DNA fingerprint using the restriction enzyme kind of techniques, you end up with thousands of different bands. But in PCR, you'd be amplifying specific genes rather than thousands of genes. You'd just be targeting specific genes. And so anyway, it can make for a simpler type of amplification, and you won't necessarily have as many bands. In some cases, again, you don't even necessarily need to use probes in some of these situations. It just kind of simplifies the whole process. But probably the best use of the PCR, again, in concert to produce the DNA fingerprint, is that you only need tiny amounts of DNA. You can amplify specific VNTRs, and it works great. So a lot of times these are used together. Okay, now there are other techniques, or there's another primary technique that's also used for amplifying or producing new copies of genes. And again, we'll talk about why would we want thousands or millions of copies of genes in a few minutes. But again, there's another technique to do this. And this other technique is known as gene cloning. And in gene cloning, what we do is actually take advantage of bacteria in their normal replication cycle to produce copies of genes for us. So we're actually going to have an organism making copies of genes for us. So let's go ahead and look at how this is done in an animation that deals with this. So you may recall when we look at bacteria that a bacterium is a prokaryotic cell, first of all, so they're small, they're usually easy to work with, they reproduce rapidly, so that's one of the advantages here. But chromosomes will have a large circular chromosome, and in addition to that, they typically have smaller loops of chromosomes called plasmids. And so what we're going to be doing here is using the plasmid to help us make copies of a particular piece of DNA. So again, we might have some kind of a target gene in our own genome, and perhaps we want to get a human gene into a bacterium so that that bacterium can make copies of that particular gene for us. So in this process then, we would go through some techniques to separate the plasmids from the chromosomes in our DNA sample. And what we would want to do then is use a restriction enzyme to make cuts. And so we want to actually make a cut in the plasmid to open up the loop. And then we want to make cuts on either side of our gene of interest so that that gene of interest can be spliced into the plasmid. Now, there are a couple of things I want to foreshadow here a little bit. And again, there are variations in this technique. But one of the problems we have at the end of this process is that we need to know which bacteria have picked up the gene of interest to us. And so, again, there are various techniques to deal with this, but in other words, we need some kind of a marker. We need those bacteria that ultimately take up this gene to say, hey, I have the gene of interest that you're interested in. It's right here. I'm a bacterium that has what you want. So in this case, what we're looking at is a plasmid that contains a gene for resistance to an antibiotic. And so that's represented here. And it also contains a gene for the metabolism of lactose. So this is what's known as a LAC-Z gene. So you'll notice that we have LAC-Z gene already indicated here, and we have this resistance to an antibiotic as well. Now, another important aspect of this is if we want to get a gene of interest into the plasmid, we're going to use a restriction enzyme to make these cuts. And as you may recall, restriction enzymes are very specific in where they make cuts. So we want to use the same restriction enzyme for both the plasmid and the DNA strand. And I'll get back to that in a second, but that's an important concept, the same restriction enzyme to make our cuts. So the restriction enzyme will be targeting specific sequences. And as the animation runs here in a second, it'll go ahead and make that cut for us, first in the plasmid. And then we'll also, again, be making cuts on either side of the gene of interest to us, and that will create fragments of DNA. Now, ideally then, some of those fragments insert themselves into the plasmid. Now, the reason we want to use the reason we want to use the same restriction enzyme for cutting the DNA strand of interest to us as well as the plasmid, which of course is also DNA, is that then it would produce complementary sticky ends. So you may recall when we talked about restriction enzymes earlier that sticky ends was a big part of that. So again, the sticky ends would be complementary ends, and then that could be inserted into the plasmid. So again, I want to emphasize the importance then of these sticky ends. So again, you may recall that if we have some strand of DNA then that we're interested in, that a particular restriction enzyme will target specific sequences, leaving these sticky ends. And then again, what we can do is use these complementary ends, and if we cut the plasmid with the same restriction enzyme, it increases the chance that this fragment will reanneal with our plasmid. So here in this case, the green is representing DNA of the plasmid. So if we can insert that in effectively, we can do that again if we use the same restriction enzyme because they're all the same kind of sticky ends, and so they're more likely to anneal with one another. Okay, so getting back to our animation then, let's go ahead and see what happens from this point then. So now we've got our gene of interest spliced into the plasmid. But what I also want you to notice is that we targeted this enzyme, this LAC-Z enzyme, and we've actually spliced the DNA fragment of interest to us into the middle of that LAC-Z enzyme. And this is part of that marking technique that I was talking about earlier. So the next step then would be to go ahead and get the plasmid into bacteria. So again, the plasmid came out of bacteria. We want to get it back into bacteria. And this is a process known as transformation that I'll talk about in a few minutes, but this is not an easy process. So a relatively few bacteria will actually reincorporate this plasmid, this modified plasmid, back into the cytoplasm. So this is why we need a marker, because again, just a tiny percentage will actually complete this process. So again, once this plasmid is inserted into the DNA, the bacterium is going to be making copies of itself and will eventually plate that out onto an agar plate to grow. And again, what we want to happen is that the bacterium will make multiple copies of itself, and every time it makes a copy, it will also be making a copy of the gene that we put into the plasmid. So anyway, what will happen then is that these bacteria will grow on the Petri plate. And again, you may recall that the plasmid was targeting an enzyme for the metabolism of lactose. And so what we would do then, or in this particular case, they're being grown on a medium, first of all, that contains an antibiotic. So if the bacteria has the plasmids, they will have the antibiotic resistant gene. So that's one thing. But they may have picked up plasmids that were not genetically modified. So the genetically modified plasmids, you may recall also, we inserted the gene of interest into the middle of that LAC-Z gene. So those that have a functional LAC-Z gene, we would put in a medium that when they're metabolizing lactose will turn blue. So in this case, the blue colonies do not have the gene of interest to us. They have picked up a plasmid, but they have not picked up a plasmid that has been modified genetically. The white colonies then would be the ones that have the plasmids and have the plasmids that have our gene of interest. So this is what I mean by some kind of a marker. We in some ways need a particular gene or some technique that will not only tell us which bacteria have picked up plasmids, but have picked up plasmids with the gene of interest to us. So that's an important concept and one that I'll come back to as we look at other techniques. So again, the idea of a marker then is to say, hey, I have the gene that you want, and I'm making multiple copies of it. So as we look at these two techniques then, PCR versus gene cloning then, One is just basically we're using an artificial cycler to produce copies, and we can target specific genes. The other, we're actually using bacteria to do the work for us, and every time the bacteria makes copies of itself, it will make copies of our gene. And then we ultimately can go back, grind up the bacteria, separate out our gene of interest. Okay. Now, why make multiple copies of genes? Well, it involves what's known as recombinant DNA technology. And recombinant DNA technology, or recombination of DNA, just means where you have unlike DNA combining together to form novel combinations of DNA. And this does occur naturally. Crossing over, for example, in meiosis is an example of a recombination where we have DNA from different sister chromatids recombining and crossing over. But again, we also have developed techniques to do this in cells. So we refer to this as genetic modification. And when we're combining genetic material from different species, we refer to this as transgenic modification, or creating transgenic organisms. So anytime we utilize recombination techniques, we refer to these as GM, or genetically modified organisms. And again, if they're utilizing, for recombining DNA from different organisms, that would be transgenic organisms. Okay, so what are some of the techniques then to get genes into cells and to genetically modify cells? One of these is transformation, which we more or less illustrated earlier. And transformation doesn't work very well, but in some cases it will. And it's a pretty simple idea. What we do is just take plasmids of DNA, we mix it with a cell. In this case, we're looking at a bacterium. And we modify the cell wall and cell membranes chemically or in other ways to make them what we call competent. In other words, we try to increase the permeability of the cell to DNA. And what we're hoping is that at least some of the cells will incorporate our plasmids, get them into the cell, and ultimately, again, what we would want to do is yield these cells and make these cells competent, more willing to take up DNA. And ultimately, we're hoping that some of that DNA gets incorporated into a plasmid or possibly into the chromosome itself. But again, we want that plasmid to somehow have a marker so that when we plate out this bacterium onto a Petri plate, that will not only have bacterial growth, but those bacteria that have the gene of interest to us will say, hey, here I am. I'm not only growing on your media, I have the gene that you want to have. So again, the idea of a marker is always really important. Okay, now another way to get genes into cells is what's known as viral transduction. So we will use a virus as a carrier to get genes into a cell. And just talking briefly about how viruses work, basically viruses, and we've talked about this a little bit I think previously, but viruses basically have the simplest viruses, and I'm talking about viruses here that actually work on bacteria, will have a shell of protein and a core of DNA. And so what they will do is attach to a bacterial cell. They'll inject their DNA into that cell, and then basically they will take over the machinery of the bacterium, utilize the bacterial DNA polymerase to make new copies of their DNA. That DNA will be transcribed, translated, and that will now produce enzymes that will generate new viruses. Eventually those viruses will be assembled, and the cell dies basically as the viruses are released into the environment. So viruses themselves don't metabolize or do work, but they take over the machinery of a host cell, and the cell then makes copies of the viruses. So again, what we can do is engineer viruses such that they will accidentally pick up a gene of interest to us rather than their own genes. And these might actually even be plasmids. I'm just representing these as fragments. And so anyway, when they attach to bacteria, then instead of injecting viral DNA into the cell, they'll be injecting our gene of interest into the cell. And then again, we're hoping that at least some of those cells will incorporate that gene into their chromosome or plasmids. And again, we also need them to somehow show us that they have picked up that gene. So we would also include some kind of a marker in our gene of interest. Now, another way to modify cells genetically is we just call direct injection. And again, this doesn't work that well, but with some cells, like large cells, ova, occasionally it works pretty well. And so, again, what you do is take a cell, and here we're looking at a eukaryotic cell with the nucleus, chromosomes inside. And so, in this situation, what we're trying to do, we literally do, is just inject fragments of DNA into a cell. We're hoping that at least some of these cells then will incorporate our gene of interest into their chromosomes through recombination. And again, we would also include some kind of a marker so that this cell says, hey, I've got your gene, I'm expressing your gene, it's functioning in me. Still another example, and this works pretty well with plants, is what we call a gene gun. Basically what we'll do here is we take tiny particles of beads, usually gold, some kind of a metal, occasionally I think glass is used, but they're put into a little gun, basically a pneumatic gun that will use air pressure to fire these pellets. And so the pellets are soaked in DNA that contains our gene of interest, and then they're loaded into this little gun, and then this gun fires into tissue specimens, and some of the beads go into cells, and there we will have the DNA separate from the beads. And again, what we hope is that at least some of the cells will potentially incorporate that DNA into their own chromosomes. And again, that DNA would need to have some kind of a marker so that the cells that have our gene of interest will say, hey, I have your gene, I'm expressing your gene. So again, none of these techniques work very well. Only a tiny percentage of the organisms that we treat in one way or another actually will take up and express the gene of interest to us. And again, there are other techniques under development all the time. These are some that are used. But again, there's new ones all the time. So every year there's something new. So I don't want you to think that that's a complete list by any stretch of the imagination. So then the question is, well, why do we want to modify genes? Why modify bacteria, for example? And again, there are a number of reasons. One is for gene cloning. We may want a cell to make copies of genes for us so that we can modify other cells. But also we can modify bacteria to produce products that are useful to us. For example, we can insert a human gene into a bacterium, get it to express that gene, and it will produce that human protein for us. It isn't necessarily just with bacteria, but we can do this with eukaryotic cells as well. And for example, this is one of the ways and one of the reasons why human insulin is used for the treatment of diabetics in modern times compared to 20 or 30 years ago. 20 or 30 years ago, basically diabetics would have to use animal insulin because it was too expensive to try to generate human insulin. But eventually, we were able to modify cells genetically, get the insulin gene into different types of cells, and have them express and produce insulin for us. So again, the human insulin that people are using today does not come from cadavers or come from other people. It comes from microorganisms that are producing this insulin for us. We can also genetically modify bacteria to make them less pathogenic. In other words, decrease their disease-causing ability. And then that's useful in producing antibiotics in some cases, but actually for immunization. So it really helps with vaccinations. And then again, there are just a million ways that we can modify and reasons we can modify. So again, we may have a lot of reasons, for example, modifying or boosting environmental activity or the physiological activity of a particular type of cell. For example, there have been bacteria that have been modified that will metabolize organic materials, basically crude oil, that have the capacity to digest crude oil. And so again, we've boosted that activity, and so when we have major oil spills in different parts of the world, these bacteria are disseminated and they help clean up this environmental mess. So again, these are just a few ways and reasons why we might modify bacteria, but there are as many reasons as you potentially can think of. And again, we can also genetically modify eukaryotic cells as well, and again, just 1,000 reasons why, but certainly one would be to produce useful products. So again, if we can modify eukaryotic cells, they can express proteins or products of interest to us. We can help, in some cases, disease. We can potentially replace defective genes, and we're still working with that, but there's been some success with that. In agriculture, again, a number of different applications. Certainly increasing crop yields in plants is one of these areas of important research. And then also, modifying cells in tissue transplantation and so on so that potentially they prevent rejection or perhaps drugs aren't needed, suppression drugs aren't needed in the amounts that they would have been needed previously. So again, there's just so many different lines of research where modifying cells genetically has some applications. Now, also organismal cloning is something that is important. And again, there are various levels in which this is done. Certainly plants are very easy to clone. We can isolate individual plant cells, strip off the cell wall, and we can isolate these cells, get them growing individual plants, and then generate individual plants from those. So cloning of plant cells is pretty easy to do. Plants are pretty easy to asexually propagate. And again, why would we necessarily need to do that? Why not just take a cutting? Well, because you can generate a lot of novel plants from just a cluster of cells. And it would be a situation where perhaps we've genetically modified a plant, or we have a plant that has unique genetic capabilities, and we want to quickly expand that population. And so this would be a technique to do that. So when you have a genetically unique organism. Now, in vertebrates, we've been able to clone amphibians for quite a while. Generally, we've used embryonic cells, merged those with egg cells in which the nucleus has been modified or eliminated, and then we've been able to get adults from that. Now, historically, we had difficulty using differentiated cells, adult cells, to do that. And as you may know, we've gotten past that hurdle, and one of the really famous example of this was the sheep Dolly. And so again, what they were able to do is actually take cells from the udder of one sheep, merge those or fuse those with an ovum in which the DNA had been destroyed. And they did that through an electrical charge. So that basically mimicked the events of fertilization. And then when the embryo grew, they inserted that into the uterus of a female and produced a new offspring that was genetically identical to the nucleus from which the cell came. So again, that's something I never thought I would see in my lifetime, but again, there is, and it's hard to do, but the reason you might want to clone an animal, again, is an animal that you have genetically modified. Because realize that every time you breed it with an animal that's not genetically modified, you're going to dilute that gene. So through cloning then, you can get a genetically identical organism to the one where the nucleus came from, and you preserve that gene of interest to you. So I'm afraid I'm out of time, so that's it till the next time.